Staphylococcus. History. It was called Staphylococcus for the first time in 1880 by Alexander Ogston, a Scottish surgeon. Isolated and provided the first taxonomic description of Staphylococcus in 1884 by Rosenbach and gave the name Staphylococcus pyogenes.
General characteristics. Gram-positive cocci. Non-spore-forming, non-motile, spherical cells, usually arranged in grape-like clusters. Single cocci, pairs, tetrads and chains are seen in liquid cultures. Can readily grow in ordinary media under aerobic and facultative anaerobic condition.
Young cocci stain strongly gram-positive. On aging many cells become gram-negative. Non-motile, non-spore-forming and non-capsulated organism. Catalase positive. Able to grow on agar contains 7 to 10% of NAC, halophilic. Grow most rapidly at 37 degrees Celsius but form pigment best at room temperature of 20-25 degrees Celsius. Colonies in solid media are round, smooth, raised and glistening. Some of them are normal flora of the skin and mucous membrane of human. Produce catalase, which differentiate them from the streptococci. Relatively resistant to drying, heat, and 9% NaCl, but readily inhibited by 3% hexachlorophene. Normal habitat. Staphylococcus are widely distributed in the environment. They form part of the normal microbial flora of the skin, upper respiratory tract and intestinal tract. S. aureus is carried in the nose of 30 to 40% or more of health people. Antigenic structure. Peptidoglycan, mucopeptide, polysaccharide, A, phosphate teichoic acid, provide the rigid exoskeleton of the cell wall. It is important in the pathogenesis. Protein A, similar to polysaccharide A, it occur in S. aureus only, unique, react with Fc region of Ig, both are virulence factor, prevent complement activation. MRSA. Although methicillin-resistant S. aureus MRSA strains are not necessarily more virulent than methicillin-sensitive S. aureus strains, some MRSA strains contain factors or genetic backgrounds that may enhance their virulence or may enable them to cause particular clinical syndromes. Toxins and enzymes. Lipase. That break down fat or lipid in the skin help organism to spreading in tissue. DNase. Destroy the DNA. Hyaluronidase. Has action in hyaluronic acid in connective tissue help S. aureus to spreading in the tissue, spreading factor. Toxins and enzymes. B. Lactamase. Antibiotic, inactivating enzyme, that lead to penicillin-resistant. Staphylokinase. Cause fibrinolysis. Coagulase. Enzyme that clots plasma and coats staphylococcal cell, which prevent the bacteria from phagocytosis. Toxin and enzyme. Catalase. Produced by Staphylococcus species used to differentiate Staphylococcus from Streptococcus. Leukocidin. Break or lysis or kill WBCs. Hemolysin. Lytic toxin, exotoxin that destroying RBCs and plat test. The three main species of clinical importance. S. aureus pathogenic. S. epidermides non-pathogenic. S. saprophyticus opportunistic. Pathogenicity. 1. Infection of skin and 2. Subcutaneous tissue. 3. Wound infection. 4. Newborn's impetigo, rash. 5. Scalded skin syndrome. Peels. 6. Secondary infection of insect bites. Lab diagnosis. Specimen. Depending on the site of infection. Microscopy. Gram-positive cocci cluster or pairs or single, non-motile, non-spore-forming and non-capsulated organism. Culture. Facultative anaerobic. 37 degrees Celsius, pH 7.2. On blood agar. Produce yellow to cream 1 to 3 m in diameter. Some strain or cis hemolytic, colony or slightly raised, smooth and shine. Lab diagnosis. Mannitol salt agar. 7 to 10% NaCl, differential and selective media for isolation or recoveries. S. aureus from fecal specimen. S. aureus produce small yellow colonies. Lab diagnosis. McConkey agar. Produce small colonies, 1 to 0.5 mm, after overnight incubation at 35–37 degrees Celsius. Most strain or non-lactose fermenter. Biochemical test. Catalase plus VE. Coagulase plus VE. S. aureus. DNase plus VE. S. aureus. Lab diagnosis. Antimicrobial sensitivity. Antibiotic with activity against S. aureus include vancomycin, cephalosporin, penicillin. Control. Avoid contamination of food. Avoid direct contact with carriers. Sterilize the surgical instruments. Personal hygienic practice.